Now, we move on to translation, the final step that leads to the synthesis of proteins. Earlier, in transcription, we moved from DNA to RNA, so we still maintain the same language of the bases. But with translation, we are moving from the language of RNA to proteins, or amino acids, hence the name translation. This occurs in the ribosome. Let's take a look at the ribosome. As we can see, the ribosome exists as two subunits in its inactive state, a large one and a small one. Let's go through the process of translation step by step. Now, before anything happens, the amino acids need to have enough energy to form peptide bonds among themselves. Remember, a protein is a long chain of amino acids linked together by peptide bonds. In order to have enough energy, the amino acids are activated in the presence of ATP and linked to their specific tRNA. This is known as the charging of tRNA. tRNA is needed to help provide the energy needed for the amino acids to form peptide bonds between each other. On to the next step. Translation begins when an mRNA comes in contact with the smaller subunit of the ribosome. Now, the protein formation has to start at the start codon, also known as AUG. This sequence is the only sequence that is recognized by the initiator tRNA. The tRNA sees the sequence AUG, and this is its signal to bind to the ribosome, thereby activating it, and to start creating the amino acid chain to form the protein. We can see that there are two binding sites on the larger subunit. Here's how the amino acid chain is formed. We start with the initiator tRNA. It binds to one of the binding sites. Now, the activated ribosomes, that is, the ones that are bound to their corresponding tRNAs, look at what the next sequence is. Here we can see that it's UUG. So we need a tRNA that reads AAC. The amino acid associated with that sequence is valine. So the tRNA linked to the amino acid valine binds to the binding site. Since the tRNA provides enough energy, a peptide bond is formed between both these amino acids. This bond is done. Now the ribosome moves down along the mRNA. The next sequence is ACG. This is related to the amino acid alanine, which has a sequence CGT. The tRNA binds to the binding site, and another peptide bond is formed. Thus, the mRNA itself doesn't become a protein. All it does is provide a blueprint for what sequence of amino acids need to be bound together to form the final protein. This process keeps going on until the ribosome reaches a stop codon. As we can see, the stop codon is UAG. A release factor binds to the stop codon and stops the process of translation. Protein synthesis is now complete, and the polypeptide is released from the ribosome. As you can see, there are some sequences before the start codon on the 5' side of the mRNA and some sequences after the stop codon on the 3' side of the mRNA. These sequences were not used during translation. They are known as untranslated regions, or UTRs, and they are very important in ensuring that the translation process is efficient. Even though the ribosomes look like tiny dots that don't do much, we can see that they aid in one of the most important processes in the cell, translation.